Pat. Hi, everyone. I'm Dr. Victoria Kirsch, the National Scientific Coordinator of CanPath, the Canadian Partnership for Tomorrow's Health. Welcome, and thanks so much for joining today's CanPath webinar, which is the first webinar to kick off CanPath's 15th anniversary. So today we'll hear from Dr. Philip Awadalla and Dr. Marie-Julie Favé, who will describe the interplay between genetic and environmental factors and their influence on chronic diseases and phenotypes, using data from CanPath, which is Canada's largest population health cohort. So Dr. Awadalla joined CanPath as a principal investigator in 2010, initially as the director of the Cardigen study in Quebec, until March 2015, and then as executive scientific director of the Ontario Health Study in July 2015. He was named National Scientific Director of CanPath in March 2018. He's a director of computational biology at the Ontario Institute for Cancer Research, and he's also a professor of population and medical genomics at the University of Toronto. And then our second speaker is Dr. Marie-Julie Favé. She completed a PhD at McGill University in population genetics and developmental biology, and then she joined Dr. Awadalla's group at the CHU St. Justine for a postdoctoral fellowship until 2017, where she applied bioinformatics and statistical genetic approaches to multi-omics data from the Cardigen cohort, which is one of the contributing cohorts of CanPath. She moved with the Awadalla lab to the Ontario Institute for Cancer Research and Vector Institute for Artificial Intelligence, where she continued her postdoc research and worked as a data science research associate. She's also been recently appointed as a faculty member at Concordia University. So following their presentation, we'll have time for questions. Please use the Q&A button at the bottom of the Zoom window to drop your questions throughout the talk, and we'll get to them toward the end. Before we start, I wanted to acknowledge the traditional territories of the Mississauga of the New Credit First Nation, Anishinaabe, Wendat, Huron, and Haudenosaunee Indigenous peoples, on which the CanPath National Coordinating Center at the University of Toronto now stands. The territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and the Confederacy of the Ojibwe and allied nations to peaceably share and care for the resources around the Great Lakes. With CanPath teams working across Canada from coast to coast, we also acknowledge the ancestral territories that are home to many Indigenous people from across Turtle Island, and we're grateful to have the opportunity to work on this land. So with that being said, we'll now get started, and I'll hand it over to Dr. Awadalla.
Thanks very much, Vicki. I'm just going to start my screen share and put it on to full view. So it's great to have you all here today. It's a beautiful day here in July in Toronto. So it's wonderful having all of you listen to us present. What I'm excited about presenting, which is our CanPath is now in its 15th year. And over the years, we've developed this fantastic resource, I feel, to support Canadian research in health. And there have been so many people, not just our participants, and we'll hear about our numbers and the data that we've captured about our participants, but there's been so many people who've contributed to CanPath's development over the years, over the past 15 years, and even before these past 15 years, in terms of getting it off the ground. So it's a real pleasure and honor to be able to present today, kickstarting if you like, our celebration of these 15 years. So today with MJ, I'll be presenting. I'm going to start things off talking about CanPath, giving you the lay of the land of where we're at in terms of building this population cohort activity, and I'll be talking specifically about one or two projects at fairly high level that I think exemplify why we need to build these types of population cohorts, not just in Canada, but in other settings across the world as well. So we've titled the presentation today, "The Genomic and Environmental Influences on Canadian Health Phenotypes and Chronic Disease Outcomes." MJ is going to be talking quite a lot about what we've been doing in the environment space, looking at genetic bioenvironment influences on these traits. As I said, I'll be talking quite a bit about the resource and how these different data assets can contribute to our understanding of early disease development among Canadians. So just this is a slide I've presented numerous times, but I think it's a good reminder of what is a population cohort and how do they differentiate or distinguish from, say, clinical cohorts. And population cohorts, if you like, are a snapshot of the population, and they're like population laboratories. We can capture, if you like, real-world evidence or real-world data about who develops diseases, and these are some diseases that are listed here in terms of their frequencies in Canadians, and who develops cancers and chronic diseases and so on. And we can ask questions related to these diseases, like how do we address the root cause of, say, health and disease in a population, and what can we do to improve our health? Can we identify communities that are more, say, at risk than others, and are these associated with, say, environment or even climate change? I'm going to be talking quite a lot today about cancer, and can we use these studies to develop tools to detect cancers earlier and perhaps be more aggressive in our interventions and so on. So today, we're going to be presenting CanPath, which is the Canadian Partnership for Tomorrow's Health, Canada's largest population health research platform. It has collected data from 1 in every 100 Canadians. And in the age range that we've targeted, this is an adult population cohort, it's effectively 1 in 50 Canadians, largely because we've recruited adults from the ages of 30 to 70 at baseline. And we started that 15 years ago. And so our median age, if you like, is shifted with, of course, the age of the biobank. And of course, we've had significant investment from many partners in the build of this program, and continue to see, and we're very grateful for that support from those investors as well. Really quickly, as I mentioned, have collected significant, deep phenotypic information, and generally the same phenotypic information from over 330,000 Canadians. In fact, we're well over 330,000 participants now. And this slide's a little bit dated because some of our numbers have changed. These are the population cohorts that make up the CanPath, if you like, family. As was mentioned during the introduction, I started off in Quebec with that build there of capturing 43,000 participants, and I'm now sitting in Ontario. But we have regional directors who operate, manage, lead both the scientific and operational activities in each one of those provinces. We started with cohorts in Quebec and Alberta, in fact, being the first out of the gate in terms of recruitment. And we are still recruiting some of our baseline participants in populations or in provinces, in Saskatchewan and Manitoba. In fact, this number could be updated because in Saskatchewan, while they're still recruiting, there are actually over 1,000 participants now have been recruited as part of the Healthy Future Saskatchewan activity. In the cartoon on the left, you see we capture questionnaire data. All of our participants consent to data linkage activities for the most part. We also have consent from all of it to be a CanPath participant, to follow these participants over time in a number of different ways, which I'll be talking about before in a few moments. We biobank many biologics from these participants. Some of these biologics get turned into data like genomics and other types of molecular features as well. This is the wonderful scientific team that we have leading the CanPath activity. I've been, as was mentioned before, I've been involved in CanPath since 2010. I co-lead at U of T, the National Coordinating Center, with Jennifer Brooks, the Executive Director of CanPath, and Dr. Trevor Dummer, who's actually at UBC, who's the National Scientific Co-Director. You can see all the individuals here who are all of our regional leads. And I actually wanted to mention that there are some of, because we're in our 15th year, there's some of these people here who've been here since the beginning. Jason Hicks, who's the Executive Director of Atlantic Path, and Trevor Dummer have been with us for this entire journey, in fact. And so it's great to highlight them. But of course, we have all of these great investigators who've also been with us now for a significant amount of time, and we're doing important work in leading this activity. So as mentioned, over 300,000 participants have completed detailed questionnaires probably at baseline when they first consent to be part of the CanPath scientific activity. From that, those questionnaires, these are fairly standard questionnaires capturing baseline health information, where we capture participant demographics, information about their health. I think we're a pretty important program in capturing prescription medication information from our participants, as that varies from province to province when trying to get that information from administrative data sources. So I think that work that we do here is very important, capturing that from a large number of individuals. And we capture all kinds of other information like anthropometric measurements, socioeconomic information, education levels, income levels, and so on. And I think we're an important cohort as well for capturing information that you wouldn't capture from clinical record data, like, say, behaviors, drinking alcohol, smoking, physical activity, and we're doing significant work now in diet as well. CanPath has collected well over 1,600 variables as a result from, say, information captured with these types of questionnaires. And we're consistently in the field. We've done a number of follow-up questionnaire activities where we capture longitudinal information from our participants as well in some of these, if you like, disease categories and other bits of information as well. We, because of our size and because of our reach into the population, we have had the opportunity to work with other important organizations across Canada to, if you like, build on the baseline that we have. And that has supported what we might call ancillary studies. These are important ancillary studies because they capture information that we may not have captured at baseline, for example, MRI data. And so we probably have one of the largest MRI collection in Canada from a population cohort, and certainly outside of UK Biobank, which has also done significant numbers of recruitment and for collecting MRI information. But one of the things that we're pretty proud of is our partnership with Sonia Anand at McMaster University in capturing MRI information and pretty extensive MRI information to support health research. Another thing I want to specifically highlight in CanPath, and I kind of touched on this when thinking about our follow-up questionnaires, is the data and the consents that we have to recontact our participants allows us to support both retrospective as well as prospective research. So because our consents allow us to follow up with our participants, we can ask our participants, how has their health changed, or how has any of their important attributes that might be associated with health has that changed over time. Our participants also consent to allowing us to link to administrative health records, which is an important component of both retrospective as well as supporting prospective research as well. We can go back into administrative health data and ask questions about, well, was there something that might have led to the development of a disease that we can capture from linked administrative health data, and we can also use those linkages to track people's health over time. And so we're very excited and proud of a new partnership between CanPath and the Canadian Institute for Health Information, where for all the CanPath participants who have consented to the linkages to administrative health records, you can now effectively, or very soon, be able to come to CanPath to capture in a one-stop shop, with the exception of Quebec, unfortunately, but at a one-stop shop, health information, linked administrative health information of our CanPath participants. Those same consents allow us to also link to environmental exposures. And so MJ will be talking more about the types of things we've been looking at with in the context of some environmental exposures and how that relates to the health of some of our CanPath participants. We're very proud of the partnership we have with the CANU program, the Canadian Urban Environmental Health and Research Consortium, which allows us to link at various degrees of geographic resolution CanPath participants to important environmental or exposome exposures. And so we've been using that extensively to look at things like, say, air pollution or the built environment and how that relates to health. Some of that was used in an important paper that was published in Nature last year, and I'm only going to touch on this really briefly, but this is a nice segue into how we're going to think about cancer in a few minutes. How some of the CanPath data and some of the work that MJ and I have done in the past was able to say something about how air pollution impacts chronic inflammation, looking at inflammatory markers, and how some of those inflammatory features are associated with the development of lung cancer in non-smoking lung cancer patients. Last thing I'm going to do in terms of our high-level overview of CanPath is just mention that because we're over 300,000 participants and we are the largest population cohort in Canada, we are one of the few, in fact, we're the only Canadian cohort that's part of the International 100,000 Cohorts Consortium, which is now called actually the International Health Cohort Consortium, which includes all of the major, if you like, uh, country-wide population cohorts like the UK Biobank, the All of Us program in the United States, and a number of other cohorts throughout the world. In fact, there are, in this database that we host here actually at the Ontario Institute for Cancer Research, there are over 70 cohorts that have over 100,000 participants where they've been able to share data dictionaries, and we've been taking those data dictionaries and looking at how those data dictionaries and the variables in them, the meta variables in them, align across those cohorts, because that allows us as scientists to be able to identify, say, participants beyond just these geographic boundaries and population cohorts and potentially build larger health research studies as well. Okay, so that's just a very quick overview of what we've been able to build and continue to build at CanPath. And so I'm going to talk specifically about a project that's close to my heart, because I feel like it's one of the reasons why we build these types of population cohorts, and what are the strengths of these types of studies. And this is specifically work that was initially started with my student Nicholas Cheng, who's currently writing up his PhD as we talk, as I'm speaking. And so I'm largely speaking about stuff that's going directly into his thesis, and some of it's been published, and some of it's is in preprint stage, and some of it's also hot off the press. But it's really in the space of how do we leverage, say, some of these great resources that we've been building in, say, CanPath or in other population cohorts in the world, to develop new tools for early detection of cancers and use those to potentially develop more aggressive interventions. So it is fairly well known that the earlier we can detect a cancer, the more effective will be our treatment of those cancers, and of course the 5-year survivability of those cancers, so of those patients. And so that's all I'm showing here in this figure at the top right. It's the 5-year survival rates as it's related to when a cancer has been diagnosed. So the stage of diagnosis is colored at the top. And all I'm showing here are the various, say, screening procedures. Many of these are very well known and developed and supported in many government jurisdictions around the world. But largely, these screening procedures are there to capture these cancers early and to support prevention before these diseases become too advanced. So in CanPath, we've been building things like the Canadian Cancer Study, where we've captured information about participants using linkages to, say, the various cancer registries in the provinces, as well as through programs like CIHI. And what those linkages allow us to do is identify participants who, say, came into the cohort healthy and, say, have remained healthy. We can also use that information to identify our prevalent cancer cases, so those people who came into the cohort with a diagnosis, and we capture that information. And of course, and make that available to researchers, and of course, identify those individuals that we call incident cancer cases. So those are participants who came into the cohort healthy and then have subsequently gone on to develop a disease. And so all that data together, this is very high level. We have much more detailed information, gives us overall, through all those linkage activities, a total number of over 41,000 as of the data as of July of last year, actually, when we finalized putting all of this data. 41,000 participants in the CanPath cohort have developed a cancer. That's only including so far actually the Atlantic provinces, Alberta, Ontario, and BC, and we're still actually, before putting this, didn't include our Quebec data. So using this information, we've gone on to try to develop what are known as liquid biopsy tools. And those liquid biopsy tools are sometimes also called or thought of as cell-free DNA tools, where we try to use, say, biobanked material, blood material, to identify or see if there was a signature in those blood samples of a cancer in this case, before that initial diagnosis. And there are many ways of looking at, say, the DNA that's been shed from tumors and being shed and found in, say, plasma or serum. And there are many ways to characterize that DNA and identify which of those how those DNA features, say, separate a potential cancer pre-patient from somebody who's cancer-free. And that's all we're showing you here. Generally, the idea is that apoptotic cells will shed DNA. And of course, there are going to be different features that can discriminate cancer DNA or DNA from cancer cells from those of normal cells. And so liquid biopsy approaches come in many different flavors, but generally the idea and the value of a liquid biopsy is that from a blood draw, one can identify somewhere in the body that there's damage, and that damage might be caused by a cancer or it could be caused by something else. But we can then develop tools. In our case, we're developing genomic tools that can discriminate, say, normal tissue from cancer tissue. And we've actually been using a DNA methylation approach to do that. And the reason we've been using DNA methylation is that different tissues in the human body show various methylation-specific signatures. And we can capitalize on that by looking at the liquid biopsy and using that methylation feature to discriminate whether what we're seeing in the plasma is coming from, say, a potential cancer or from a tissue versus that what we'd see normally in blood. And so we've been developing these tools, and we've been using resources like the TCGA to develop this in a pan-cancer kind of way. And we've been using resources like the Ontario Health Study, where we have 40,000 banked plasma samples from the Ontario Health Study participants. And we've tracked over, using those data linkages that I just mentioned a moment ago, who gave us a blood sample and then has gone on to develop a cancer. So when we get that information, say, from the Ontario Cancer Registry, we can go back into the biobank, pull those plasma samples, and they may vary from, say, 7 years before they were actually diagnosed to maybe a few months from when they were diagnosed. And that's what this picture is trying to show. Say, for breast cancers, you get these varying ranges in terms of time since somebody gave us a blood sample and we banked it, comparing that to when they were diagnosed. So we have a lot of those for breast cancers, and this was actually our first poll when we started this activity back in, say, 2017 or 18. We have a number of prostate cancers. We don't have as many pancreatic cancers, and that's because the incident rates for pancreatic cancers aren't quite as high as that of prostate and breast. And when we do these polls, we also can see that we get information from, say, the registries about stage of diagnosis when they were diagnosed. We also have information about things like grade, and we have some information about survivability as well. And you can see that for breast cancer, for example, the figure with all the pink histograms is that most people in Ontario, when they are diagnosed, they're diagnosed at stage 1. And that's, again, because of the breast screening protocols that we have in place in the population. That same can be said for prostate cancer, but less so for, say, pancreatic cancer, where it seems to be more advanced by the time it's actually diagnosed or detected. So we see more stage 2, stage 3, stage 4 for pancreatic cancers there. So these are the numbers we started with on the right, and we used, again, as I was saying before, a liquid biopsy approach which used DNA methylation as a way of identifying features that might be associated with, say, breast tissue or breast cancer tissue versus that of normal tissue. And what you can see here in our first analyses and with these principal components is that in the colors where we have our cancer participants is that this approach separates fairly well participants who were diagnosed with the cancer versus controls for both breast, prostate, as well as the few samples that we had for pancreatic cancer. Taking this further, we went back after that poll, and this is now where we're at in terms of numbers. So you can see there are sample sizes for breast cancer alone are significantly larger. We did another poll, and so we've doubled our incident breast cancer cases. And you can see here that not only are we capturing information about when the blood was stored and biobanked and when they were diagnosed relative to that blood, but we also have information about when a participant had a mammogram. And so that allows us to also ask whether that mammogram detected the cancer or whether the liquid biopsy detected the cancer and the mammogram didn't detect it. And so we've been using that, capturing that information and evaluating those questions. And so generally, we're not only able to see very quickly, I'm going to not get into the statistics too much here, but we use a tenfold cross-validated approach for it's something of a machine learning approach to determine our overall accuracy. And generally, we do fairly well. We were around 75% accuracy in our first pull of the data. And now with our second pull, we have enough data that we can also look at how well can we do based on subtypes and or age of diagnosis. And I was actually really quite surprised is that our areas under the curve for cancer detection for participants diagnosed before the age of 50 are remarkably high, actually, compared to participants who are diagnosed later in life. And that might speak to the fact that those are actually maybe slightly different diseases, or they are different diseases. So the areas, our ability to detect a cancer seems to highly depend on how old you are when you were detected. Subtypes, triple negatives, our areas under the curve for triple negatives are very high compared to other types, as well as time of and subtypes. We also do really well in the context of, sorry, these are just some numbers, looking at this in the context of when we're actually detecting cancer, and sometimes we're doing extremely well compared to mammography as well. How are we doing for prostate? Well, for prostate, we're using slightly different features coming from the genomic material, and generally we're doing okay. We're around 70, our areas under the curve are around 78%. And I'll just highlight that what this does highlight is a challenge in these spaces, because there are potentially participants in our cohort who might actually have a cancer, but we're defining them as controls because they hadn't yet been defined or diagnosed with the cancer, but we don't know that yet. And so there might be individuals in the cohort where our sensitivity and specificity are impacted largely because we're doing this in a retrospective sort of way. Okay, and I'm just lastly going to say that, yeah, these are some, in the case of metastatic cancers, these tools do extremely well, and that's largely because these are much more advanced cancers, and when a cancer is metastatic, of course, it's shedding much more material, and the signature is much stronger. Okay, so just to kind of, the last thing I wanted to just kind of highlight here is that these tools are actually not only good at detecting things like cancer, but they also can detect other damage or other insults, maybe due to other types of environmental exposures. And one of the things that Nick and I have been studying with this data is our ability to see other exposures like alcohol and the impact of alcohol. In fact, actually, the alcohol signatures in plasma in the context of cell-free DNA are actually remarkably strong and striking, in fact. And so this is something we're going to be following over time. So just to summarize, large population cohorts can enable pre-diagnosis profiling. All those linkages to administrative health data really support our ability to do this in a safe way. I argue that this type of work is being done more safely here because these people are already being treated, the cancers have already been detected, and we're just going back into our biobanks and seeing if there was a signature that we can use. And there might be other signatures that are coming from other information or questionnaires that might help us improve our ability to score and identify participants in early disease settings. As I said, we can see signatures, particularly for breast cancers, up to 7 years prior to diagnosis. Generally, we're using a methylation program to be able to do this. And I would argue that these liquid biopsy approaches should or could be at least accompanying other kind of traditional screening approaches in the population as well. The last thing I wanted to highlight before I turn this over to MJ is that while we're working in that cancer space, these population cohorts are really valuable for also thinking about healthy aging. Who are our participants in our cohort that are aging remarkably well? Who are our 90-year-olds in the cohort who say might have a blood profile of a 40 or 50-year-old as well? And I'm not going to have time to talk about this today, but my student Alyssa Bader, who graduated last year, actually was using much of the material that we have in the Ontario Health Study specifically to identify those participants who we might consider healthy aging based on, say, things like blood profiles, linkages to administrative data, and so on. And she was able to also develop her own sort of risk scores for doing that using this type of data. So this is just a shout that you can use our information if you would like to build different kinds of risk scores based on the epidemiological data. design studies in a way that allows you to look at, say, things like healthy aging. So who are our participants who are aging particularly well? Who are participants who are young and not aging well? And identify features that might be associated with healthy aging. And this is the last thing I'm going to show about this type of thing, is that we were able to show that with the bloods that we have frozen and biobanked, we can support single cell applications as well. And so that Alyssa's project is largely focused on using these single cell approaches to characterize healthy aging using her experimental design. So that's all I'm going to really say there. I just wanted to thank all of the funders and our partners across Canada who support the CanPath activity. This is just a list of the names. I also just wanted to thank Nick specifically for the work that I presented today, and that's him right here. And I'm going to now then turn this over to MJ.
Thank you so much, Philip. That was fantastic. It's great to hear about all these efforts that have been made into this fantastic cohort of CanPath. And it's been really an immense privilege for me to be able to work on this resource since almost the very beginning of when the genomic data was being released, and to see where it's been going so far. So this is to show you how a little snapshot of the genetic diversity that CanPath now has in terms of data. And this doesn't even include the last release of the Carthagena data, which has been completely genotyped. We just didn't have time to include it in our own data sets, but you see that across Ontario and Quebec. So those are PCA plot based on genotypes of the participants. So each little dot is a participant. And we've added on top of that the 1,000 Genome Project, so we can have an idea of the ancestrality of the participants that have been genotyped in CanPath. And while the majority of them, of the participants, they cluster in the space where participants are identified as European ancestry, there's more and more diversity in the newest release of genotyping data, which include more participants from Africa, from Asian ancestry, and from admixed American ancestry. And this is great because it's been an issue and a challenge in human genomics in the last couple of years that was claimed to be addressed to really increase the genetic diversity in populations cohorts. So as Philip described, the biosamples, CanPath is primarily collecting blood for the participants because usually humans are not really willing to give anything else than blood when they're still alive. But blood is a fantastic tissue because it goes around the body every day and it's replenished by new cells fairly often. And this process of hematopoiesis really creates a very complex and dynamic tissue that is in touch with all your organs and also all your environmental exposure that people are encountering during their life. And that makes a really great snapshot to study health, and both in terms of the genomic basis and the environmental basis that is channeled through the immune system. So one study, so I'll start with a study that we published several years ago, but it's one of the early study we did with the beginning of the release of the genomic and transcriptomic data in CanPath, where we selected participants in 3 different cities across Quebec that is so the Carthagena cohort is an interesting population because it was founded by well, I'll go over that in a few slides, but it captured the demographic history of the particular colonization of the Quebec province from the French settlers. But also, like, those 3 cities represent also very distinct environment where Montreal is a very large urbanized city. Quebec is a smaller city, and the Saguenay region is a smaller urban environment, but is also home to a lot of industries, which of course affect the environment that the people over there encountered every day. So for this particular study, we used blood from 1,000 participants from those 3 different cities, where we performed genome-wide genotyping. So for that particular study, it was just 100,000 participants. Now there are almost 70,000 participants that have been genotyped across CanPath. But what I think is really exciting about these participants, these thousands early one, is that we have bulk RNA sequencing from the blood. So it's really how the blood functions and how the immune system reacts and functions in that particular environment. And we also use the extensive health questionnaire, history and physical measures on the participants that makes CanPath so rich. And also linked to the CANU data set, where we use data to characterize the exposure of these participants. Mostly like the geographic exposure linked to their physical environment where they actually live. So as I touched upon 2 minutes ago, the Quebec population is super interesting to study because it's a founder population. So Quebec was colonized from, I mean, there were people living here already, but like the European descendants are coming from the French population that colonized first Quebec City. And then some of these settlers, and then they went into found Montreal. And then some of them went up north to colonize the Saguenay-Lac-Saint-Jean region. So this created a series of population bottlenecks, and we can observe and confirm that in today's genome of the descendant of those settlers in the French-Canadian population, where those types of bottleneck actually affects genetic diversity across the genome. And we could actually, using the genotyping information, we can tell apart people that are of French-Canadian descent versus people who are more, their close ancestors are from the French population or European population. But we could even go a bit further, because we could even tell apart people within the French-Canadian population that are originating from a certain subregion because of the sequential bottleneck. So we could tell apart whether our participants have a regional ancestry, mostly coming from the Saguenay region, from Montreal, or from Quebec City. And there's previous and subsequent study that have shown that the French-Canadian genes, the genetic diversity, mirrors Quebec geography and demographic history of sequential bottlenecks. And I just listed a few here. So this is just to show that we could actually do that in our 1,000 participants. So if we look at this, this quantification of genetic diversity in terms of principal component, and we plot that visually on a map, you can see really that, you know, the color represents how the principal component, and you can see across Montreal, Quebec, and Saguenay that the participants are quite different from each other. In parallel, we also perform in changes across the 3 different cities, which is quite striking and interesting, because what drives gene expression kind could be either the genetic ancestry underlying that, because people are slightly different and they might express genes slightly differently. Or it could be the environment that is also slightly different that might modulate this expression. So the question now is this gene expression modulated by genetic ancestry or by the shared environment of these people? So we perform a range of comparison, like using, as Philip said, like this population as an experimental laboratory, which I'm not going to get into because it becomes a bit long to explain. But like we looked at whether how many genes we could see that are differentially expressed across the different combination of people living in a city and people having a genetic ancestry from that city or from a different city. And what we found was a striking pattern of a bunch of genes. Like in that case, we selected 170 by the end, for which we see the expression was very highly correlated with the habitat that the people would live in. So either they would live in Quebec, in Montreal, or in Saguenay, but it wasn't associated with our genetic ancestry at all. So the regional ancestry, or even participants that are coming, that were inferred to be from European population, where when they live in a city, for example, if they live in Saguenay, their gene expression across those genes were more similar to people living in Saguenay than from people living in Europe. Right? So basically, our gene expression clustered by environment and not by ancestries. And what was interesting in the first step in trying to ask what's driving this pattern is that there was an enrichment of genes that were involving gas transport and exchanges in inflammatory pathways. And this is just to know, this is something that's been observed in other cohorts where your geographic origin drives your expression or your methylation on your genome more so than your immediate genetic ancestry. And this has been observed in Morocco, in Fiji population, and in Indonesia. So to look a little further into what could be driving this pattern, so I worked with Dr. Fabien Lamas, who was a postdoc in the lab at the time, and we designed this pipeline to look at the to combine the gene expression data and the health questionnaire or health physical measure of the people to see, can we actually find variables measured on the people that are associated with these changes in gene expression, as well as changes in the environment as well. So what we found is that regarding the environment, where we cross our gene expression with the KNU data set, we could find that like what really makes those gene expression apart across city wasn't really the city, it was really the air composition measure. So the small particulate matter, NO2, and particularly sulfur dioxide emission 2 weeks prior to the blood draw. This was quite interesting because those changes are very striking when you look at the population in the Saguenay region, where there's a much higher SO2 exposition because of the industrial emissions. And what we find as well is that in these people that are more exposed to these mixture of pollutants, they have a higher arterial stiffness, which is like the rigidity of your arteries, and the stiffer they are, the worse it is for you in terms of cardiovascular health. So this was higher with high SO2 exposure, and it was correlated with changes in gene expression that we measured. It increases the risk of cardiovascular diseases, and is also associated with asthma and lung diseases, which also have been found to be quite prevalent in the Saguenay region. So to continue on that topic, we also designed a subsequent study with a fantastic PhD student. I graduated a year and a half ago, Michelle Hardwood, where she, instead of looking at gene expression per se, so the actual level of expression, she took the Carthagena cohort and she looked at, for particular position in the genome, the what we call allele-specific expression, which is the imbalance between, so we all have 2 alleles at a gene, so one coming from the dad, one coming from the mom, and the way the gene is expressed when the RNA is done, it could be maybe the paternal allele will express more than the maternal allele. And this is an interesting phenomenon that's been observed across the tree of life, and it's also associated in humans with particular conditions. So she looked at allele-specific expression across our same 1,000 participants, and she wanted to ask whether where does she find this type of changes in allele-specific expression, and whether it was associated with any kind of diseases. So what she found was a very interesting pattern where she could infer using recombination pattern in the genome that allele-specific expression could buffer the effect of deleterious mutation. So for example, if someone has a deleterious mutation at a particular location, so it would increase the risk for certain disease, if the actual allele of your gene is not expressed as much, that would help to not develop that conditions. And what she found is that in population like in Quebec that have a smaller population size because of the demographic history of bottlenecks, they see that this phenomenon of buffering deleterious mutation is not as efficient. And then she also went to do a second study looking at the effect on different conditions, including type 2 diabetes, cardiovascular diseases, and cancer. And one thing that I thought was super interesting that she found is that medication response, not if you take the medication or if you're sick, but your response to the medication, for example, if someone takes statin because they have a condition that like a dyslipidemia that they need to be on statin, then they see that the medication response to statin is associated with particular allele-specific expression pattern at certain genes. So another very interesting data set that came out of CanPath a bit more recently was the cytokine profiling in immune cells on about 8,000 participants across the Canadian landscape. So what I show you here, the map of the Canadian map, is whenever you see a color, don't worry too much about the color, but the actual color is where you have participants in that neighborhood that have been profiled for cytokines. And thanks to Elias in the lab who performed the experiment, then we can look at like the level of those cytokines in the blood of the participants and look at whether they respond to or they're associated with certain exposures. Cytokines, they have diverse function in the immune system and beyond. They promote inflammation, they can help to fight infection, injuries, and stimulate the production and activation of certain immune cells. And this is just want to highlight how beautiful this data set is, because you can map the participants to really like the 3-digit postal code level and really start looking at like, is there a particular environmental exposure that might drive certain patterns in those cytokine levels which are associated with immune functions. So what we did as a little pilot project is to look at whether we could reproduce those effects that we found in the gene expression that are associated with high pollution exposure. Can we find similar effect in the associated cytokines level? In that case, we have way many more participants to look at. So we've looked at NO2 exposure and PM2.5 exposure. And what you see here on the graph or on the y-axis, so each dot is a different cytokine. On the x-axis is the effect, so whether it's overexpressed in high pollution or underexpressed in high pollution. And on the y-axis is the significance level, so the higher it is, the more significant the association is. And what we could find is that we did reproduce some of the patterns that we found with the gene expression. So certain cytokines, we had the matching gene in the gene expression. And for example, interleukin 8 that you see on the graph on the left, that's highlighted in blue. I don't know if you see my mouse over here. It was also seen, so that cytokine is overexpressed in participants that are exposed to high NO2, and this was similarly overexpressed in terms of the gene expression. So we have a nice little addition to our pollution story. And I'll touch upon a bit on this, what Philip started discussing about this, some single cell data that we generated recently with this fantastic student, Elisa Bader, which I think I have her picture on the next. Yeah, Elisa, with the help of Elias, who performed all the lab work, and our fantastic bioinformatician, Mausi and Vanessa. So Philip touched upon a bit where we could use CanPath to study how people age. So some people age in a very healthy fashion and some people age not so well. So we designed this experiment where we performed single-cell RNA sequencing on 400 participants. And I want to mention, this is really great because this is one of the largest human single-cell healthy aging, but also just healthy population cohort on which there's single-cell data available. And this is an incredible data set to analyze in so many different ways. So we could capture changes in cell number in certain subtype of cells with people that are healthy agers. And one thing that we wanted to look at as well is to match this single cell expression data with whole genome sequencing and genotyping, in that case, whole genome sequencing on those participants to find expression quantitative trait loci. So those are SNPs in the genome that are associated with the expression of a nearby gene. So for example, you have 3 genotypes at a certain position in your genome, and each of genotype is associated with a certain expression level of your gene. With an increasing number of derived allele, then you get an increasing level of gene expression. But this can be modulated by environmental exposure. So for example, oops, sorry, I clicked the wrong button. You can think that you have such relationship in individuals that are of high risk of having a complication in the health versus people who are healthy agers, and they might have a different relationship between the SNP and the gene expression level. So for those who are interested in the actual modeling of it, this is the type of linear model that we perform across all the different cell types that we have, across all this, across all the genes that are expressed in any cell type. And we found a bunch of them. Is a different little dot you see is a particular SNP, a particular position in the genome where we found that like the reaction, the gene expression level was associated differently depending if you're a healthy ager or non-healthy ager, indicating that these might be contributing or associated with the downstream effect of healthy aging. And interestingly, they're also located in certain particular chromatin region where we mix that, we overlap that with an attack sequencing data set that we perform on these similar participants. And we replicated a certain number of these associations using the Cartaja and bulk RNA sequencing data. So to summarize, like this has been a really great journey to work with an increasing and more complex data set on CanPath, where now it includes bulk RNA sequencing data, single cell RNA sequencing data, cytokine levels. We also contributed in those massive consortium efforts for example, the Global Biobank Meta-Analysis Initiative that developed polygenic risk score that could eventually be used in the clinic to predict the risk of any individual to develop certain diseases. We've also contributed to the EQTL Gen Consortium by doing meta-analysis on those EQTL loci across different cohorts and ancestries. So really, CanPath is an incredible resource where you can have not only genetic data, but also all these omics and exposure data set where we can really start to design, tackle a lot of questions like predictive disease modeling, or really understanding the effect on the environment, not only on the phenotypes, but really on like the actual function of the cells themselves. So the same pictures Philip showed, it's like a massive like team effort that's been done over the year, and it's been incredible to have the opportunity to work on that. And I really wish a great future to CanPath and more studies and data generation.
Great, thank you so much, MJ and Philip. We do have a few questions that have come in. Richard Gould is asking, have you explored whether there are any biomarkers or genomic markers that may be associated with increased risk for substance use disorders? I know you presented, yes, some data on methylation patterns being correlated with alcohol consumption, but maybe you can say more.
Yeah, yeah, that's directly. Yes, actually, there was a study published using some of CanPath data in a meta-analysis looking at genetic predispositions, and it was a paper that came out in Nature Genetics. We're just one of many cohorts that was used in that, largely around risky. It was considered risk. It was called risky behaviors. And so there has been some work in that space. I wouldn't say it's been very in-depth and detailed though, but I think that's really the only one that comes to mind right now, looking at, say, a GWAS type of approach or genome-wide association study approach.
Great, thanks. Monica Perry says, thank you for your presentation. I'm just wondering if race and ethnicity are captured for all people in the database. And what gender variables are collected? Identity, roles, relationships. So I could probably answer that one. Yes, we do collect both race and ethnicity. And then the gender variables, it sort of evolved a little bit over time and varies a little bit between the individual cohorts, but we do have a comprehensive list, a section on sexual orientation and gender identity. We ask about biological sex. We ask about whether they consider themselves to be trans. We ask about assigned sex at birth, felt gender, and gender that they currently live in their day-to-day life. So it is sort of a comprehensive resource. Okay, Yan Wei is asking, thanks so much for the excellent talks. Regarding genetic data, UK Biobank has partnered with Genomics England and has 500,000 whole genome sequenced. Wondering if there's any plan for CanPath to partner with Genome Canada, for example, to get more participants' genome sequenced.
Yeah, and there are lots of discussions in that space. And so we have some discussions going on right now with specific partners in terms of helping us generate more genomic data. We didn't get really into specifically, like MJ talked a little bit about what we've done in Ontario and Quebec, and she meant Quebec she mentioned, has already genotyped all of its cohort, and that coupled with imputation information is a pretty valuable resource. Ontario is about over halfway through genotyping all of its cohort, and then we have snapshots of genotyping in BC, Alberta, and the Maritimes right now. So overall, altogether, that means that there are about almost 60,000 participants in CanPath where there are genotypes available. It's not as comprehensive, of course, as genome sequencing. And so, yes, we're having those types of conversations, and we've been having conversations with Genome Canada as well about, numbers, participants, where would the data go once it's been generated as well. So.
Thanks. Okay, someone is asking, how can this amount of detail help me address our priority populations? I mean, and I think, I can answer that. I think, your populations that you're interested in can be defined on, by various methods, by, the self-reported data that we've collected, which includes ethnicity, education, socioeconomic status, or through some of maybe the linked environmental data. So we have data linked to, pollution, exposure to pollution, postal code, so those could be sources of data sort of to, subset your data or look for interactions by specific populations of interest. Okay, someone's asking, what environmental exposure metrics are available in the CanPath data set? So as I just mentioned, so we've linked CanPath participants using postal code to data connected by Canoe. And Canoe collects data on, environmental data on air, noise pollution, green space, climate change, socioeconomic status, for example. And actually, this question will probably be answered in detail in a recent sort of office hours section that CanPath is hosting along with Canoe. It's scheduled for July 23rd at 1. So if you're interested in learning more about the Canoe data, that's sort of a great opportunity to ask more questions.
Vicky, there's another question that's popped up on the Q&A.
Sure. So this one's from Helen Doyle. Do you collect indoor environmental exposure data or proxy indicators? Example, housing conditions, mold, indoor overheating, access to air conditioning. I don't believe they're captured through Canoe.
So that's right. I think some of them are not. And some of these are being captured. Well, we do. And Canoe does capture some of these proxy indicators. So for example, I remember seeing some information or studies where they were looking at arsenic, say, exposures as it relates, and mostly captured through geographic location as sort of as a proxy. We've had other ancillary studies supported by other agencies in Canada where things like radon captured in households have been supported. So that's being done. But I would say that's being done not at a CanPath wide scale, but more at a project by project or regional level as well.
Great, thanks so much. Okay, so we're at 3 o'clock now. Thank you, everyone, for joining. Feel free to join our access hours. Check out our celebratory 15th anniversary video on our website. And thanks so much. Take care.